We were the Intercropping Field Experiment Group that focused on crop growth and yield. This experiment consisted of 4 treatments: corn, squash, and beans intercrop, corn monoculture, squash monoculture, and bean monoculture. Our research topic was intercropping affects crop growth and yield because of interspecific competition, mutualistic species interactions, and spatial intensification of production. Our learning objectives were to compare monocropping and intercropping in terms of crop growth and yield, and to observe whether or not interspecific interactions, in addition to intercropping, affect crop growth and yield. In order to understand our learning objectives and to explain how we think crop growth and yield will be affected, our group focused on the following variables: total harvestable biomass, root depth/volume, canopy via the string technique, height/growth, nutrient status, and marketable yield.
Lily and Yahaira were the biomass group which focused on tracking the change in crop biomass during the trajectory of this project. We did this by taking 2 types of measurements: the fresh and dry weight of our above ground biomass. Our methods were random samplings of 2 plants for each crop in each plot, totaling in 36 crop samples for each harvest day. For each of the 3 harvest days, the materials needed were large paper bags, Sharpies, shovels, a scale, an aluminum tray, cutting blades, and a drying oven. After locating the samples in the field, we took steps to ensure that the samples were not marked by other groups. We then removed the entire plant from the soil, including its roots, and placed it within its matching paper bag. Once all the samples were collected, we removed the roots at the soil line and measured the fresh weight of each sample and recorded the data in our table. These samples were then placed in the drying ovens by our TAs at 65 degrees Celsius for 48 hours. The dry weight was then measured and recorded.
Rufeda and China measured plant height. First, we used a random number generator to develop a numbering system for corn and bean plants in a plot. This helped us randomly select 5 plants per plot that we measured for the duration of the quarter. We created a datasheet and input our data via smartphone. For ease and speed of measurement in the field, we marked plants that would be measured by spraying the soil of the chosen plant, as well as a small dot of spray paint on the leaf for increased visibility. Every alternate week, we measured plant height from the soil to the highest point of the plant with a meter stick and recorded height in centimeters on the datasheet. Some corn plants died due to the spray paint, so we measured the plant right next to it down the row instead.
Francine and I looked at the roots of the monocrop versus intercropped corn and beans. Our hypothesis was that the monocropping system was conducive of a shallower root depths and less biomass, and the intercropping system stimulates a deeper root depth and greater biomass. The metrics that we looked at were the root count, root angles, and fresh and dry root mass. At harvest, we collected 1 bean and 1 corn sample from each plot of each treatment. We washed the roots and soaked them to loosen the soil, agitated them in soapy water to remove the soil, and then rinsed them clean. The tools we used for measuring were a shovel, a thumbtack, 5-gallon buckets with and without soapy water, scissors, a utility knife, the Maize Shovelnomics scorecard, and paper bags.
To test our hypothesis, Vanessa and I counted all root types except for branching roots. This included basal roots, tap roots, brace roots, and adventitious roots. The root count helped determine if intercrop plants put more energy into growing a greater root biomass. Next, we measured root angles using the Shovelomics scorecard. By measuring the root growth pattern at 2 angles, we hope to determine if the roots were preferentially growing away from the neighboring plants or growing at a non-discriminant pattern. Finally, we cut the roots off the shoots at the soil line to measure the fresh root mass. The root samples were then dried in a 65 degrees Celsius oven for 48 hours. The dried root biomass was recorded. The fresh and dry biomass measurements were taken to help determine if more energy was focused on growth of the roots as opposed to shoot growth.
We measured the canopy index. Canopy index is pretty easy to measure. All it takes is a weight attached to a string and some markers. We used wooden stakes as markers, but flags or other markers would also work. After we transplanted the crops into the field, we went through the field and randomly planted markers into the plots. We planted 5 markers in each plot to make for a total of 60 sample sites. We assigned each sample site a unique identification number to efficiently organize our data.
We dangled the weighted string from above the marker so that the weight made contact with the marker. We recorded the number of points of contact between the crop canopy and the string. We repeated for each individual sample site and repeated these measurements for all sample sites every other week.
We measured chlorophyll content as a measure of nutrient concentration of our plants. To do this, we measured 5 plants from each of the monocrop plots and 5 bean plants and 5 corn plants from the intercropping plots. Each week of data collection, we used an apogee chlorophyll meter to take 3 measurements from a leaf on each of the sampled plants, then average those 3 measurements to get a single value of chlorophyll content for each plant.
For marketable yield, our methods were to develop a numbering system for plants and each crop species in a plot, then randomly select 5 plants per crop species per plot. Then on harvest day, we harvested the edible part of the plant and placed it in a labeled container. Once the edible part was harvested, we weighed our fresh sample on a balance and recorded fresh weight in a datasheet. The materials we used were orange containers, a utility knife or a box cutter, a balance, a pen, and a datasheet.
Measuring the canopy index allowed us to quantify photosynthetic capacity. The higher photosynthetic capacity of the intercrop treatment relative to the monocrop treatments indicates that intercropping systems utilize more of the available sunlight for production. Since sunlight is an infinite resource, there's no trade-off to using as much as possible.
A major challenge we faced was that some of the corn plants we had marked died. This was because of the use of spray paint on the base of the recently transplanted corn plants, which ended up killing a few of them. To overcome this challenge, we used the plant next to the dead one. But at least now we know for next time that the spray paint has the potential to kill. Furthermore, it should be noted that some of the surviving corn that were spray painted seem to look a little shorter than the rest of the plants in the row, which could affect our results. For plant height, we expected the monocrop system to be taller than the intercrop system due to the presence of interspecific competition within the intercrop system. Although we observed by the end of our experiment that the average monocrop corn was actually just slightly shorter than the intercrop corn. On the other hand, intercrop bean was much shorter than the monocrop bean as predicted.